So I'd like to take us on another fun little journey. The work that you did over at Chernobyl. You sussed out the fact that isotopes could be stored in mycorrhizae fungi. And then those damn Russians drove into friggin' Chernobyl and dug in and set up shop and stripped every one of those friggin' goddamn nuclear alarms because they broke the fungi. So can you tell us a little bit more about what you found during that work, that incredible work that you did?
We found that some of the plants growing in the field that had mycorrhizal fungi growing inside of them. And so maybe I need to talk a little bit better about what that process was. The root system on the carrots. Yeah, so you have the carrot itself, which is orange and fairly chunky. The green top up here, but then hyphae growing out from the root of that carrot. And every one of those samples that we looked at, where there was mycorrhizal fungi coming from one plant and colonizing the next plant, and colonizing the next plant and the next one, none of the actual carrot root had the
Radioactive
radioactive materials. And so where are those radioactive materials? And we took the root systems, the small, on the sides, we took that material off and looked at it, and whoa, that was, guess what? That's where those radioactive materials were being stored by the carrots. And so, we didn't have money to go looking at more kinds of plants that had, were they, like blueberry. You need to have a you need to have a mycorrhizal partner to that as well. And so would it be the same? Would blueberry, blackberry, whatever, would it have those problems in the berries? And as long as they had mycorrhizal fungi, I believe they would. Now, we didn't actually look. Where we had the carrots and they had been treated with some sort of weedicide on it, and it killed the fungus, then the root of the carrot had radioactive material in it.
Amazing. Just amazing. And it just proves that what you discovered there was in fact true when they went in and drove their tanks and ripped the whole friggin' forest up, because they released all that. So obviously a lot of other plants were doing the same thing, is basically saying, no, I don't want that isotope. So you go bury it over somewhere else, in someone else's backyard. What is it, NIMBY? Not in my backyard.
There's been a group in England that has been going around the world looking at these different kinds of interactions, and their data fits our data perfectly.
Nice.
So it's getting out there to the wider era, to the wider people audience. Yep. And it's we've got some work that we want to do this not this summer. It's a little too late. The money got in too late. But next spring we'll be looking at that kind of situation a bit more in depth. So
would it be just isotopes, or would it be all kinds of different things like heavy metals?
Yeah, we plan on doing work with heavy metals, all kinds of different toxic chemicals as well as the yeah.
It's funny, but again, Elaine, you have inspired so many people across the globe in so many different directions. And again, I want to thank you from the bottom of my heart, because it's given me the ability to live a life that feeds my soul. I'm not chasing money, but I'm doing good things that make me feel good. And I don't know if you remember this project that I had with James Satillo. It was the Chattanooga auto dealership. And I came to you and I said, Elaine, I had this oil tested. It's got a tremendous amount of coal tar. And you said, all right, you need Pseudomonas. And I'm like, okay, well, how do I make Pseudomonas compost? And you warned me. You said, Leighton, if you're sick, you're going to get really sick playing with this stuff. But if you do it appropriately, Pseudomonas will, and you respect it, the Pseudomonas won't hurt you. And you gave me a recipe. I made it up. I forget, I was still at Rodale or now, but I made this compost and sent them down the extract. And sure as shit, they cleaned it up. So, I mean, again, bioremediation is like going to be one of the real big issues if we don't crash into the 2030 agenda, all this shit, right? But there are so many Superfund sites and so many dirty areas. Even, just a regular farmland has so many heritage freaking chemicals, atrazines. And so, this is really like the one of the most important areas that we need to look at in the future, is that through composting correctly and appropriately and using materials that we know do have these specific bacteria that can break these metals apart, or these compounds, degraders and decomposers and solubilizers, we can make amazing changes. And in a short period of time. I mean, we did that in 1 year early. It went from horrible levels of coal tar to very little in the course of about 18 months. And so have you been playing around with any more of that type of bioremediation work?
We've got a proposal that looks like it will be funded, maybe, hopefully, please. And if
fingers crossed.
Yeah, how many ways can you cross things?
Do you remember Twister?
So if we get that funded, then we're going to be using compost as a filter for all that contaminated water to go through. And it should be possible. We should be able to manage it. You got to realize that, gosh, I'm not going to remember the correct number of digits in here, but it's like 6,000 new toxic chemicals are released each year.
Mm-hmm.
On top of all of those that we've already had. So how many, since World War I, how many years has that been? And we've been happily producing new and different toxic chemicals that are soluble. They move into the water. They move into your wells, your lakes, your oceans, and no one seems to get upset by this. We have to stop trying to grow our own food using toxic chemicals. It's just such a mental, what are you thinking about?
Money.
And that's all that matters in this world, right? Is money.
It appears like that sometimes, Elaine. And the reason I asked that is because a good friend of mine, Leaf, has gotten a job down in Oklahoma studying mine waste. So this is a lead mine. And the wastewater that's coming out of there is horribly toxic. And originally, they set up a series of bags filled with spent mushroom substrate only, and filter the water through that and change the bags periodically. And they have had a tremendous amount of success with just that. And I believe he's hoping if he gets allowed to continue in the program, to start introducing more compost, because we did a study with Furman where he reached out to me. He had shown that the ability to break atrazine down to secondary and tertiary metabolites using white rot fungi, but the metabolites were just as toxic, but they didn't last as long. So he wanted to eliminate them. So he asked me if my BioComplete compost would maybe benefit it. And I was like, heck yeah, why not? It can't hurt, right? So they did a series. Say again?
there have been papers on atrazine showing that in a very short period of time, they could be completely decomposed.
Yeah, yeah. So when they added the compost with the white rot fungi, gone.
Yeah.
So again, it drives back to the fungally complete or fungally dominant compost as the solution to the pollution.
Yep.
It's not dilution. It's to remove it.
Right.